All right, welcome everyone. We're group number 3. We are starting lesson number 13, the group problems. These were a little difficult for us. I'm sure it's difficult for everybody else taking the course. Hopefully we can provide enough explanation that these will make sense to you. We're going to start off with Sophia going through number 1.
Okay, so we have, it says you wish to use DNA sequencing, the Sanger method, to determine the nucleotide sequence of the DNA molecule shown below. So 5' and then TAGTC, whatever, primer annealing site, and the 3' end. So the primer annealing site indicates the region of the template DNA where the primer will anneal during the sequencing reaction in order to generate the newly synthesized strand. Note that the newly synthesized strand will be complementary to the sequence above. After DNA sequencing using the Sanger method, described in class, the DNA fragments were separated by electrophoresis on a gel. And so the first part of the question asks, what would the binding pattern look like on the gel? In order to answer this part of the question, I suggest, so you have your little electrophoresis, and you have your walls on one side, and you have your columns of the DDGTP, DDATP, DDTTP, and DDCTP. And so they're going to be at different lengths, and just determining on where like the base lies, it's going to fall. So basically, since there's a G that's closer to the primer annealing end, it's going to be higher up. Does that make sense? Am I explaining this correctly?
I think so. I think you're synthesizing the DNA inside of a test tube. You're adding your DTGTP, which is going to stop your DNA
yeah, so it's going to make the sequence like shorter.
Yeah.
So for every time it hits the complementary base, it's just going to get shorter and shorter, and you're going to have every single time there's supposed to be a G added, you're going to have like a longer or shorter base.
Yeah.
It's going to be like that for every single DDATP, DDTTP, and DDCTP. And this is just what like the electrophoresis looks like.
Yeah.
So then the next part, B asks, suppose I use E. coli DNA polymerase 1 during the sequencing reaction instead of the Taq polymerase. So basically, it just wouldn't work because the temperatures are too high and it would just be denatured. And then C asks, suppose I accidentally add E. coli DNA helicase to the reaction in addition to the normal DNA sequencing reaction components. And it's just not going to affect it at all. And then suppose I add DDATP to one of the 4 tubes. However, I forget to add DGTP, DDTTP, and DDCTP to the other tubes. Assume that the other components using for the sequence are normal. So the Taq polymerase will not have DDNTPs to use to make the DNA sequence, so only the DDATP will be bound to the annealed DNA sequence, and only the DDATP would show up in the gel. So none of the other ones would even show up because they're not in there in the first place.
Yep.
All right. And I have number 2. For letter A, it says during a typical PCR cycle, the DNA synthesis step is done at 72 degrees Celsius, while the primer annealing step is done at 55 degrees Celsius. B is AMP-R is a common selectable marker that is found in many plasmid vectors, while the restriction endonuclease recognize and cleave DNA molecules at planedronic sites. C, during agarose gel electrophoresis, DNA is placed in an electrical field. DNA migrates toward the positive electrode during electrophoresis. D, DDATP lacks the 2' OH group and a 3' OH group. Transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. And F, during blue-white screening, IPTG is commonly used to indicate the expression of the lac operon, while XGAL is cleaved by beta-galactosidase to turn E. coli cells blue.
All right, so I have number 3. This one is you're interested in PCR to amplify the region of a double-stranded DNA fragment from E. coli. Then it gives you the sequence to be copied. It's just located between the 2 bold sequences. So the first thing is asking us to write the single-stranded DNA sequence that we'd use for primers in the PCR experiment. So we use, we can use, right here we have the 2 bottom ones right here. As we read from 5' to 3' for the first one right here. And then on the other side, we're also going to read 5' to 3'. So that's how we're reading it on the bottom strand right there. And that's going to be the primer for the top strand. The other one's going to be the primer for the bottom strand. And then for B, it's the equation used to determine the temperature that should be used for PCR. So what we're going to do, it gives us part of the equation. We have to find out the amount of GC nucleotides in it and the percentage sequence. And so there was 8 in the first one we have, and then 9 in the second. So you do 8 divided by 16, 9 divided by 16. Got Alicia's daughter in there now. So 8 divided by 16, 19 divided, or 9 divided by 16. Get the different percentages. And then after that, so one's 50%, and then 56.25%. Then you minus it by the 675 divided by the amount of nucleotides we have in our sequence, and 16. And you take that all together, you get 59.8 degrees Celsius for primer 1 and 62.4 degrees Celsius for primer 2. And then the next one, it's talking about if what are the sequences of the 2 newly synthesized strands if DDCTP is accidentally included in the PCR experiment. Assume that DATP and DTTP and DGTP and all the other appropriate PCR components are found in the reaction mixture. So what happens is, if DDCTP is included, we're going to go down the strand we are copying, right? Copy, yeah. And once we get to a C, it's going to stop, and then it's going to. So that gives us our first segment down there, and then for this, then it would do the same thing again until it gets to another C. Then once again, it'll stop. So I think I'm doing okay. This one's kind of hard, but
yeah. The hard concept to get, because you're going down the primer is going to go down this line like this, and the first C it hits, which is going to be the G here, and it's going to stop right here, and then that's the length there, and then the 5' to the other end. It's going to hit your first C, which is bonding to an G, so it's making a T, going to bond an A, going to bond a T, and then once it hits the C, it stops. And the same thing goes the other way.
Okay. And the last one is when we learn about the DNA replication E. coli, the DNA and DAM, why these 2 proteins not included in the PCR experiment, because they are E. coli's way of copying DNA. And so for the PCR separation of DNA is being done by a high temperature. So, and the TAC doesn't require, it all it requires is primers instead of the proteins. So.
Yep. And so I've got number 4. So number 4 is going to be a little bit more difficult to try and explain. I hope we do this good enough job. Pretty much it's a mapping technique. We're going to map out what's going on here. And so it says pretty much it gives us these 6 reactions. And it introduces BAMH, EcoR1, and HINDI3 to a same linear DNA molecule. And so in each case, you get these DNA fragments coming off. Cool, awesome. then the part A says draw an image that represents the result of the agarose gel of electrophoresis. Also include a marker that contains these markers on it. And so we're going to take each one of the reactions 1 through 6, and our marker, and we're going to pretty much make a gel, or what it looks like, what the gel would look like as soon as you're done. So we're going to have the reactions, and then we're going to have these pretty much bars or X's. I just made them X's, but there's going to be little bars on each one of these for each one of the different reactions, which gives us our different length of base pairs throughout. So cool. And then part B says draw a linear restriction map that indicates the location of the restriction enzyme sites along the hypothetical DNA molecule. This requires a little bit of trickery. It's kind of a puzzle. What I do is I looked at the BAMH1, EcoR1, and HinD3. I looked at the base pairs of each one and saw that it's a 900 base pair length linear DNA, and that the smallest ones were, or that because BAMH had 3 fragments produced, that it's going to have two spots, or 2 BAMH sites on our linear DNA site. And then from there, I knew that EcoR1 and HinD3 were somewhere in the middle of those two. And so because EcoR1 had 200 base pairs and HinD3 had 300 base pairs from the end, so I knew that EcoR1 came first, HinD3 came second. This is how I personally solved it. And so EcoR1 came first because 50 plus 100 equals 150. Or I think it says hundreds.
Might want to label those base pairs, maybe. So
yeah, I probably should. I think this is 150 base pairs, actually, because EcoR1 is 200 from the end, so that's 150. This one's supposed to be 100, I believe. So that's 200 EcoR1, 300 to HinD3, and then from the back, you have EcoR1 is 700. So this should add up to 700 from EcoR1 to the back end. So 500 plus 200 is 700. And then the HinD3 is 600. So HinD3 would sit right here. 500 plus 100 is 600. And so this would be your hypothetical DNA map. And then the last one is, what are the predicted fragment length of this linear DNA molecule is cleaved with all 3 molecules simultaneously? They're right here, 50, 100, and 500. So that's the answer to that one. I hope that made sense. So we're on to number 5. Eliza.
Sorry, I forgot I was on mute, so I was ready to go, but I was muted. So I'm at the grocery store with my nieces. Sorry, let me get back to where I was. Okay, so number 5. So for step number 1, you're going to generate your favorite gene's cDNA. For that step, you're going to A, isolate YFG's mature mRNA, B, add a poly-DT primer for reverse transcription, C, make an RNA-DNA hybrid molecule, D, add RNase H that will cut out the RNA part of the hybrid, E, add DNA ligase and DNA polymerase 1 to complete your favorite gene cDNA. 2, clone a cDNA version of your favorite gene into a plasmid vector. A, for that one, the restriction enzymes from before cut out the cDNA in the plasmid. And B, at the sticky ends of each of those hydrogen bonds are formed by putting the cDNA into the plasmid with the help of an added DNA ligase. And 3, blue and white screening to identify plasmids that contain the YFG, your favorite gene cDNA. So A, you're going to grow the E. coli, and some will have successfully transformed with the YFG inserted into the plasmid. B, put the E. coli onto agar plates with IPTG, ampicillin, and XGAL. C, after they grow, white colonies were successful transformations with recombinant DNA molecules, and blue ones didn't contain any. And D, DNA sequencing or restriction mapping can be used to verify which recombinant DNA molecules contain your favorite gene insert. So I don't know if that made sense, but those were my answers. So there you go.
All right, well, that's a wrap. Hope you enjoyed the semester and all of our mini videos that we've had here together.
From all over the country.
all over the country. Missouri, California, Texas, and Idaho. 2 of those were from one person. So, anyway, have a good night. See you later.